right, dudes. So now let's look at, now that we've covered what our carbohydrates are, let's look at how a forensic chemist would test for these carbohydrates. And this is really interesting, and it's actually fairly important to the work because quite a lot of organic matter is carbohydrates. We'll start talking about proteins and DNA in a future lesson. So once she has the organic samples from a crime scene, how would the forensic scientist, how would she identify what they are? Well, basically, you want to be able to explain that by the end of it. And when we say explain that, we essentially mean you want to be able to explain how it would work with the equations, okay? And also you want to be able to explain in terms like what a reducing sugar is, what a non-reducing sugar is, and why some sugars are reducing sugars and why they aren't. We're actually going to learn a few new terms this lesson. So, starting with aldoses and ketoses. So, here we have glucose and here we have fructose. Glucose and fructose, when they are in aqueous solution, they exist in equilibrium. You can tell because it's got this rad symbol there. They exist in equilibrium in both an open chain form and a closed chain form. So as a ring and as a chain. So when the ring opens up, you either get an aldehyde group, okay, or you get a ketone group. So aldoses are sugars with an aldehyde group in the open form. Ketoses are sugars with a ketone group. Now, an aldehyde group or an alkyl group is this one here. It's right on the end. See, there's the H there. For a ketone group, it's in the middle somewhere. It can be any 1 to 6. Oh, sorry. In this case, 2 to 5. But it's not on the end. This is called a carbonyl group, by the way, just in case you didn't quite remember that from earlier. And that's a phrase we use a lot, a carbonyl group. A carbonyl group is when you've got the C with the double bond to an O. That's a carbonyl group. Okay, so an aldehyde has aldose. An aldose has the carbonyl group on the end, and a ketose has the carbonyl group in the middle. All right, and as it says just there. So sugars with open rings, they're able to be easily oxidized. Now here we get the oxidation reducing jargon, and it gets confusing. So remember this: sugars with open rings, so an open chain sugar, can be easily oxidized by a mild oxidizing agent. Now that's important. I actually have to get back. You have to get rid of that. So a mild oxidizing agent means that it is very easy to oxidize. So we call it reducing sugars. They're called reducing sugars because they reduce the oxidizing agent. So even though they are oxidized, they cause the oxidizing agent to reduce. Okay, so they are reduced, they are oxidized, which means they are reducing something else. So sugars can be oxidized by mild agents. Mild oxidizing agents are called reducing sugars. Non-reducing sugars are not able to be oxidized by mild oxidizing agents. You can still oxidize them, but not with mild oxidizing agents. The carbon where the active carbonyl group is the carbon where the active carbonyl group is located is the anomeric carbon. So it's very reactive in the open form sugar. So the anomeric carbon. So right here, that's the anomeric carbon. That's the anomeric carbon. Glucose is a reducing sugar, and it's oxidized to carboxylic acid, as is fructose. It's also oxidized to a carboxylic acid. See, it's just the formula is a little bit different. We can use this to distinguish amongst carbohydrates. So it allows us to break it into smaller groups. And we know that the carbohydrate we found at the scene of the crime is related to this one. It belongs in this group. Okay, so here's a quick little summary. That we're only looking at monosaccharides so far. We've got glucose and fructose. Here's the structure. You need to remember those. You kind of need to know them. Here's the open structure. Again, there is our anomeric carbons. One is at the end, one is in the middle. All right, so what is the glycosidic bond? Now we're talking about disaccharides. So a disaccharide, such as this is maltose. That is the glycosidic bond. It's right in the middle there. It is the one that links the monomers together. So it's this glycosidic link that determines the reducibility of a disaccharide. So most of our monosaccharides are reducing sugars, and pretty much all of the monosaccharides you will deal with are reducing sugars. Disaccharides, they may or may not be. It depends on whether one of those sugars, one of those monomers, can open up into an open chain structure, which gives it isomers. So maltose and lactose, they are reducing sugars. Now, the reason they're able to do it is because this here, the anomeric carbon is not on the glycosidic bond for both of them. So this one here will not break because the anomeric carbon is part of that glycosidic, it's on the same one as the glycosidic bond, but this one can, and we'll show you right here. So this is our maltose. Its anomeric carbons are, well, potential anomeric carbons are there or there. In this case, though, you've got another one that's over here. Because this oxygen is attached to the glycosidic bond, it won't break apart, but this one will. So you've got the C1, so this is your 1 over here. That's also 1, but one of the C1s is not, so the carbonyl group, the glycosidic bond does not go through both of the carbonyl groups here. So it goes through this carbonyl group, like that can form a carbonyl group, but it does not go through this one over here. So it's all fine, it's all hunky-dory. Sucrose, on the other hand, is a non-reducing sugar. Neither of its rings can convert to an open chain form. You can't get that double bonded carbonyl group because we'll pop this up. Because the linkages are, because the linkages between, so the glycosidic link is between, but through what both carbonyl groups would be. For example, this carbon here would become one of the carbonyl groups, and this carbon here would become one of the carbonyl groups. So if your glycosidic link touches that bond there, like that's adjacent to the joining with that oxygen, it can't happen. So here it can't happen, and here it can't happen. Whereas up here, the glycosidic link is there, so that would be drawn just as a straight line the way it is there, or down, whatever. That's not touched to it, so it's easier to do. So that's basically reducing disaccharides and monosaccharides. Now how do we work it out? So there's two ways to do it. We've got Benedict's solution. It's a deep blue solution of copper sulfate in an alkaline citrate. Don't stress about that just yet. So it needs the alkaline condition for the oxidation to occur. And when the reducing sugar is warmed gently, so if it's a reducing sugar, it'll turn to a brick red copper oxide as opposed to just blue copper ions. So we start off with the nice and blue over this side. Here's our sugar. So it's Aldehydes Ketosis. You need to know both of these. You need to know this equation, and the next one's going to come up too. So we can see you've got a blue here, and in a reducing sugar, it becomes copper oxide. So what we get is this brick red color. Then we've got Tollens reagent. It's a colorless solution, and when we put it, we mix it with, we warm it gently again, and the oxidation occurs with silver. For example, it starts off colorless here, and you get this silver precipitate forming on the edge of your test tube. And this is a quick breakdown of the common disaccharides. Okay, so it's 2 carbonyl atoms. It's not, so glycosidic link goes through 2 carbonyl atoms, so it's not going to make an open chain structure. Whereas maltose and lactose, they do. Yes, carbonyl atom there, carbon there, but not right there. And yes, carbonyl carbon right there, but not there. So with sucrose, we'll have no reaction with either of those, but with maltose and lactose, we do. And there are other ways to test those apart. Now, if we want to test for starch and glycogen, so these are our other types. So if we want to work out if we've got a plant carbohydrate or an animal carbohydrate at the scene of a crime, we come over here. So we can do the iodine test where we add iodine to the starch and we warm it up. Now, sometimes if we've got a really strong solution, you won't, and that's what you've done in the past in class. You've had a really strong solution. You didn't need to warm it up at all. But if you have a very dilute solution, you will need to warm it up. And it'll make this purplish solution over here. So that's starch and amylose, or amylose is the version of starch over there that reacts to that. Glycogen, on the other hand, with iodine, it turns a reddish brown. And so that's what we're dealing with here. So that's glycogen. Now, just very simply, forensic chemists, she would rarely use these simple qualitative methods as an end point, as an end method. These would be used very early on to, like she would use these to just have, she goes, here's my sample, do a very quick test with it. All right, it's a reducing sugar. I need to go down this road here. I need to do these further tests, which we don't talk about, like mass spectroscopy, stuff like that, chromatography. We'll talk about them a little bit, but like just not right now. She then moves on to those further methods to identify. So this is like an initial test for your working forensic chemist. All right, I'll see you in class.